In earlier class, we studied about the development of the endosperm. It was a major post-fertilization event. After the formation of the zygote undergoes a resting period or a dormant period. The endosperm starts developing. So this is to ensure proper nutrient or ensure nutrient to the developing embryo. So today we will talk about the development of the zygote into embryo, which is known as Embryo Genesis. The zygote undergoes its own cellulosic cell wall and then divides transversely. So let's see how it divides. This is the zygote, which is 2N. Now the zygote will divide transversely. We will take this as the micropylar end and this as the chalazial end. We are taking orthotropous ovule. Otherwise, the commonly used ovule, the common ovule which is found in angiosperm is anatropous, which is inverted upside down. So the zygote is always found at the micropylar end of the embryo sac. Now this will divide transversely. This is, every time we have to mention that this is the chalazial end and this is the micropylar end. The cell towards the micropyle is known as basal cell and the cell towards the chalazial end is known as terminal cell. Terminal cell. This is also known as epibasal cell. This is known as epibasal cell. The basal cell is also known as hypobasal cell. So you can write it here as hypobasal cell. This is hypobasal cell. Now the basal cell will undergo transverse division. This is going to undergo transverse division and form. Let's see what it forms. This is the terminal cell and this forms, undergoes, the basal part undergoes transverse division and it forms many cells. This is going to form many cells. That means a long stalk-like structure which is known as suspensor. The end cells of the basal, the last cell of the basal will get little rounded up. So we call this as hostorium cell. Hostorium cell or the cell sac. This is also known as cell sac. This is suspensor. This hole is known as suspensor. The function of the suspensor is to push the embryo deep into the endosperm so that it gets ensured nutrition. Recall that endosperm is a reserve nutrition. Now let us understand how the suspensor pushes the embryo deep into the endosperm. Let's draw an embryo sac. The zygote, here I am taking the anatropous ovule. This is the micropylar end and this is the chalazial end. The zygote is here and these all are endospermic cells. This is endospermic cells. The zygote here will develop the suspensor. A suspensor is made here. The suspensor pushes the embryo deep into the zygote, into the endosperm. Now, the function of suspensor is now clear. Its only work is to push the embryo deep into the the endosperm so that it gets assured nutrition. The terminal cell or the epibasal cell will now divide along the planes to form a quadrate and then a octet. Let's see. This is the suspensor, ostomium cell. Now this divides to form a quadrate. So you can say it is a quadrate. Quadrate means 4 cells. In this condition, it is known as proembryo. This is known as proembryo. So this is the terminal cell. This is terminal cell. This proembryo will again divide and form a octet, means 8 cells. It will form a octet. Now we can't see all the cells dividing because it's a ball-like structure. Some cells will be in front of us and some at the back side. But still we are drawing it. So this is an octet. So in this condition, it will be known as globular embryo. This is known as globular embryo. A periclinial division takes place which gives rise to dermatogen which develops afterwards into epidermis. So this is the suspensor and this is the sac cell or the ostomium, and this whole is the suspensor. The cell next to the proembryo, the cell of the suspensor which is next to the proembryo, I'll change the color here, this one, will develop into hypophysis. Hypophysis. This hypophysis will divide 8 times and give rise, the 4 cells will give rise to epicotyle, not epicotyle but hypocotyle and radical. The next 4 cells will give rise to epicotyle and the plumule. So all these are arising from the hypophysis. Now the globular embryo, the cotyledons, they are growing. The embryo is growing. So it will spread little and get inclined towards the side and develop into heart shape. Let's see how it develops. It will develop into heart-shaped embryo. So this is the embryo. This is the suspensor and this is the cotyledons. The plumule is placed here. This is the plumule. So this is heart-shaped embryo. This is heart-shaped embryo. heart-shaped embryo. The cotyledons are growing. Now the cotyledons will grow and get inclined to just get the space. So it gets inclined towards one direction. This is the suspensor. And the cotyledon, the embryo is growing. Like this. Okay. So this is the plumule. Two cotyledons are coming out. That means it is a dicot embryo. So cotyledons are this. Cotyledons. Then above the plumule is the epicotyle. Above the epicotyle is the hypocotyle. And the radical is placed. Radical is just opposite to plumule. Now let us try to understand the basic structure of an embryo. We have taken dicot embryo. The point from where the cotyledon arises is known as the embryonal axis. This is known as embryonal axis. The embryonal axis, just above the embryonal axis, is the epicotyle. The epicotyle rises up and gives rise to plumule. Plumule is geotrophic, negatively geotrophic. It gives rise to the shoot apex. Now coming down, the cotyledons, just below the cotyledon is the hypocotyle. The hypocotyle gives rise, if we just come down the hypocotyle, we will see the radicals. Radical is there, which is geotrophically positive. And below the radical is the root tip. The root tip is covered by a root cap. So this was the whole structure of the dicot embryo. Two cotyledons are present, and the point from where the cotyledon arises is known as embryonal axis. So in today's class, we have seen how a dicot embryo develops. This is one of the major post-fertilization events, the changes that are taking place in the zygote, then developing into proembryo, proembryo developing into globular embryo, globular embryo developing into heart-shaped embryo, and then it is giving rise to horseshoe-shaped embryo. Then we have seen the different inclination at which different parts of an embryo is formed. In the next class, we will talk about the monocot embryo. The difference is dicot embryo has got two cotyledons and monocot embryo has one cotyledon.